Hello, everyone. My name is Angela Solis, and today I will be talking about the different mechanisms of DNA and the different stages that DNA goes through. And so to start off, we know that the DNA is categorized into threes, but we're gonna start off with the leading strand and the lagging strand. And the leading strand is synthesized to continuously go along the 5 to 3 inches, and the lagging strand is for 3 to 5, and those are in short stretches. So for the leading strand, it's also known as the Login strand, which was named after a Japanese scientist, and this is because polymers can add nucleotides in one direction. So each requires a primer to start the synthesis, and it's synthesized by pole with this little symbol. And continuing on, the importance of telomeres is the end of the linear chromosomes are protected, and the transcription often covers more than one gene and produces polycitronic mRNAs that specify more than one protein. So, in in reality, this is the distinctive structure that is found at the end of our chromosomes. So, this consists of the same sequence of bases repeated, and they serve as the three major purposes. One being it helps organize each of our 46 chromosomes in the nucleus, and they help protect the end of our chromosomes by forming a cap. So, without the telomeres, it's important to understand that DNA would be lost every time that a cell divides. So it's important for it to keep a cap. And transcription is the process of making of RNA a copy of a gene's DNA sequence. So mRNA carries the gene's protein information encoded in DNA. We also have the alternative splicing, and this is a process during the gene expression that allows a single gene to code for multiple proteins. And this is also known as a molecular mechanism that modifies pre-mRNA and constructs prior to translation. So we know that the differences between introns and ectrons, I'm going to go over that right now. And for introns, it's found in the eukaryotes, and the DNA basis is found in between exons. So introns are removed in the nucleus before the mRNA removes to the cytoplasm. And in as for the exons, it's found in both the prokaryotic and the eukaryotic, and these are the coding areas of the DNA. And exons are highly conserved DNA bases that are translated into proteins. Going into introns, these are the intervening sequences between two exons found in eukaryotes, and they do not directly code for proteins. They are removed before the mRNA forms the proteins, and it's essential to remove introns and to prevent the formation of incorrect proteins. As for exons, the coding sequences that code for the amino acid sequence of the protein, and they are present in mature mRNA after the post transcriptional modification. These are highly conserved sequences. They do not frequently change with time. And for translation, the anticodons and the codons is the difference is that an anticodon has to deal with which type of RNA is carrying it. As for the tRNA, also known as transfer RNA, it is an essential component of translation, and the main function of tRNA is to transfer an amino acids during protein synthesis. The three tRNA sites are labeled P, A, and E. The P site is peptidyl site blind, the tRNA holding the peptidyl sites. The A site is the acceptor site. It binds to amino acyl tRNA. And the E site serves as threshold. It's the final transitory step before tRNA, and now bereft of its amino acids, is to let go by the ribosomes. Thank you. This concludes my video.